And good, thanks, Hernan. Yeah, welcome to the first seminar this semester. Today it is remote, but next week we will hopefully be back in person. And actually, next week's speaker is Rich Müller. Many of us know him. And the title of his talk is going to be From Dinosaurs to Nuclear Waste, an Adventure in Physics. And I want to really stress that Rich is looking forward to do this in person. So I hope as many as of us can or dare to attend his lecture in person. Yeah, let's come to today's speaker, Hernan Garcia. Hernan is actually an assistant professor since 2015 at UC Berkeley in the molecular and cell biology department, but also at physics. Hernan studied physics in Buenos Aires, and his thesis topic was the development of pulsed solid-state lasers for nonlinear microscopies. He then did his PhD at Caltech on quantitative and systematic dissection of transcriptional networks, and did then his postdoc at Princeton. In 2015, he became an assistant professor here, but he holds now also appointments at the Marine Biological Laboratory at Massachusetts, is a co-director there for a course on physical biology in the cell, and also at Cold Spring Harbor Laboratories, where he is also an instructor for a similar course. And he was also active at the Kavli Institute for Theoretical Physics in Santa Barbara, where he was a co-director for quantitative biology. Hernan is also author of the book Physical Biology of the Cell, and received a prize from Biological and Biosciences for this textbook. Yeah, Hernan also received really a number of accolades as a Dicker Fellow for his postdoc, Burroughs Wellcome Fund career, also for his postdoc. Alfred Sloan Foundation is well known to the physics community, the Herman Award, but he's also received an NIH Director's New Innovator Award and NSF career. And recently, he is now a Chan Zuckerberg BioHub investigator and has also won the Elizabeth Hay New Investigator Award from the Society for Developmental Biology. Today, Hernan is going to talk about the breaking the standard model, but not the standard model we all know so well, physics, but that of embryonic development. And I'm looking forward to hear more about what that standard model is and how it's broken and what that means for science. Please, Hernan.
Right. Can people hear me?
I can hear you.
All right. Okay. So I wish we were in person, but that's okay. Well, let's make the best out of it. And so today I'm going to walk you through the work we've been doing over the last 6 years in my lab. And I want to give you a sense of, sorry, let me just turn on the pointer, laser pointer, there you go. What I want to do today is give you a sense of the great opportunities in dissecting and the great challenges in understanding living matter. And in particular, I'm going to walk you through some work we've been doing to establish the fruit fly as a substrate for physical biology and how these efforts led us to break what I call the standard model of embryonic development. All right, so one of the reasons I was drawn to physics is its like very clear definition of success, which at least I define as predictive understanding, being able to predict the result of experiments before you actually do those experiments. And there are many really cool examples from the study of inanimate matter. Think of the standard model where we can predict particles before we even measure them. Think of the fact that we predicted the shape of gravitational waves originating from the merger of black holes before we detected those, or the fact that you can measure a shift in a solar spectrum of 1 part in a million and predict that a planet is orbiting that star. So in that context, what we're curious about asking is whether we can reach the same type of predictive understanding in the context of living matter. And for example, could I understand how molecules come in and out to create these little tubules that separate the DNA into daughter cells, or how cells talk to each other, work together in order to create a multicellular animal, of how a bunch of fish, a school of fish, is trying to escape a sea lion that is trying to have them for lunch. So in all these questions, what I'm interested in answering is, can the interplay between theory and experiment that is so familiar to physics in the context of inanimate matter shed light on the fundamental rules behind the emergence of structure and order in living matter? And in particular, in my lab, we ask this question in the context of embryonic development. And the way we go about answering this question is very much inspired by the classic approach in physics. Meaning, for example, here's one example that I from classical physics that I love, which is what you see on the left are the results from Petit and Dulong that measured the specific heat of a bunch of solids. They found that all those specific heats were the same. And this wasn't that trivial to explain. It took Maxwell some time to figure this out using equipartition. And everything was fine until this guy Weber came along and thought of a knob, the temperature. And he showed that the heat capacity changes with temperature. And as a result, that, and the equipartition could not explain this. And this led to a crisis, and this led to Einstein proposing the quantum theory of solids. So can the same approach be brought to the study of living matter? In that case, what are the knobs that we should tune both experimentally and theoretically? And what are the precision measurements that we need to make? So the talk today is going to be finding about these knobs in the context of fruit fly development. And these knobs, their meaning will become clear in a few slides. But let's just say that it has to do with the copy number of the different molecular players that turn on and off genes, where the DNA is accessible and such. Okay. So the talk is going to be divided in a few very short vignettes. Okay. The first thing I need to tell you is give you a little bit of an introduction to the biology of cellular decision making, just so that we're all on the same page in terms of the phenomenology. Then I'm going to show you how we use the dialogue between theory and experiment to enable physical biology of embryonic development. After that, we're going to talk about the standard model of cellular decision making, which is rooted in equilibrium statistical mechanics. That model is going to make predictions that, in order to test, we're going to have to actually invent technology that didn't exist before. And once we put our theory and experiments together, we're going to find a knob that reveals that the standard model doesn't work. And that's going to lead us to thinking about non-equilibrium models of cellular decision making. And it's going to lead us to a new exciting round of the theory experiment data. And please, if you have questions or for clarification, please stop us along the way. You can write in the chat, or you can unmute yourself and ask the question. If it's like bigger picture stuff, maybe that's something that we can leave to the end. All right. So I keep talking about cellular decision making. What do I mean by that? Remember that in your bodies, you have 10 to the 14 cells. Each one of these cells has 2 meters worth of DNA with the same information encoded in the language of A, T, C's, and G's. And yet, even though every cell has the same information, some of them decide to become part of the brain, some are part of the blood stream, and some are part of the skin. So how do these decisions come about? In order to understand this, you need to know something about the so-called central dogma of molecular biology. And here's the idea. As I said, the information is encoded in the DNA in the language of genes. These genes are read out by a molecular machine called RNA polymerase. The name is not that important, but the point is that it's like a Xerox machine. It binds to the DNA, it moves along the DNA, and it makes a copy of the gene in the language of RNA. This RNA is then captured by another molecular machine called the ribosome that translates the language from the RNA into a protein. And proteins are the molecules that encode for action and for the identity of the cell. So first approximation, think, for example, of hemoglobin that transports oxygen throughout your body. Okay, so it turns out that the main game for deciding whether a cell is going to have one fate or the other has to do with turning on and off the production of specific proteins that endow them with identities. And the way the production of these proteins is turned on and off is, for the most part, by regulating the access of the RNA polymerase Xerox machine to the promoter. So the Xerox machine needs to land on a landing site called the promoter in the vicinity of the gene. And what happens is that in brain cells, brain genes will have their promoter landing sites available, but skin genes won't. And the inverse will be true in skin cells. Okay, so this is the main game in town, regulating the access to the promoter. But how do you implement this at the molecular level? Well, the last few decades of molecular biology have revealed that the central dogma is not linear, that instead there's feedback. There are proteins that feed back into the central dogma. There are activators and repressors. Activators recruit the polymerase Xerox machine to the genes. Repressors inhibit the binding of the polymerase Xerox machine on the gene. Okay, and therefore they can turn on or off genes. Okay, so so for these little cartoons, let me give you a little bit of a vibe, of a sense for the action of these activators and repressors. So I'm going to show you some data, and the technologies behind them are going to become clearer later on. But just suspend disbelief for a second. You're going to see a bunch of cells with a little red spot. That red spot indicates that the gene is on. I'm going to add repressor, and it's going to be labeled in green. And repressor concentration is going to grow. You're going to see more green. And at some point, there's going to be so much repressor that the gene is going to be coming active. Okay, so check it out. Green is growing. Dots are off. Let's look at it again. More repressor, more repressor, more repressor. Dots turn off. The same thing can be said for an activator. Here, the green is the activator as it grows. Bam, there you go. Now you get your red dots. Okay. So, all right. So hopefully, I give you a sense of how cells make decisions. Again, by turning on and off genes through the action of these repressors and activators. What I want to do now is go beyond these little drawings that I showed you and show you how we actually how we implement the physical biology approach to try to turn these cartoons into theoretical predictions. Specifically, what I mean is the following. If you open the pages of any textbook on biology and you look for activation, for example, for the most part, you're going to see a cartoon model like this one, where you're going to see, well, an activator binds to the DNA, the Xerox machine is also bound, they interact through some interaction, and that increases the rate of production of RNA. We're going to turn these cartoon models into precise mathematical statements that make predictions. For example, that predict an input-output function, how the activator concentration dictates the rate of RNA production. And then we're going to test these predictions by developing technology that allows us to control and measure the activator concentration and to measure the rate of RNA production in individual cells. And the point is that this is a dialogue. Okay, so theory is there to make experimentally testable predictions. Experiments are there to test the theoretical models and to inform the next round of theory. Okay, and we put this theory experiment dialogue to work in the context of how a single cell in an embryo of a fruit fly leads to a multicellular organism. Okay, so let me give you just a little bit more biology here of how you actually go from one cell to an animal. Okay, and flies, just like us, start with a single cell, and the first job of those cells is to make many copies of themselves. Those cells then start making movements, making structures just that result in the segmented body plan of the fly. Okay, so these segments, these are the equivalent of 2 your vertebrae. Okay, and these segments then are endowed with an identity, such as, are you going to be a leg segment or a wing segment or an eye segment? Okay, once again, if you look at the developmental biology textbook, you're going to see these little cartoons of snapshots. But one of the amazing things about development is how dynamic it is. And one of the fantastic things about flies is that we own their genome, meaning I can edit the genome of a fly such that every cell in the embryo is fluorescent. I can put the 0.5 millimeter length embryos under the microscope and watch the process of cell proliferation in real time. Okay, so here you see how cells are going through multiple rounds of synchronized divisions. And even though at 3 hours, all the cells look the same to you, each one of these cells has already decided what it's going to be when it grows up. Because each one of those cells is expressing a unique combination, contains a unique combination of activators and repressors. And those decisions are manifested by these massive rearrangements called gastrulation, where you see, for example, the separation between the head and the thorax of the embryo. The movie below takes over when the movie above left off. And all I want you to see is, as you look around the periphery of this embryo, I want you to notice the segments appearing. Okay, so notice the repeated structure. Now there's this process called germplasm retraction. And what you're going to see is that this thing looks exactly like the cartoon that I showed you in the previous slide. This is at 10 hours after fertilization. You wait a few more hours, and this thing starts twitching, because at 24 hours, a larva is going to come out of it. So in 24 hours, you made an animal. So this is an amazing substrate for asking the types of questions that we're interested in. Not only that, over the last few decades, people have figured out the combinations of activators and repressors that endow each cell with their identity. And they've discovered that these activators and repressors are organized in a cascade, meaning there's a first layer of activators and repressors with these funny names that are expressing shallow gradients. Each cell reads out that combination of repressors and activators, and each cell, and this leads to the production of a set of other activators and repressors in broad domains that leads to genes expressed in 7 stripes, that gives you genes in 14 stripes, that gives you the segments of the embryo, okay? And we don't only know the molecular identities of these activators and repressors, we also know their connections, meaning this guy activates this guy represses this guy. So by any measure I can think of, the fruit fly is almost like the hydrogen atom of developmental biology, meaning it's a system that is simple enough that I could dream of having a system of equations that describes it, and yet it captures the essence of much more complex developmental processes. But I have a complaint, and that complaint is that despite this amazing state of the art, just like this is not a pipe, this is not a fly. What I mean by that is, if I changed all the names on this network and I give it to the world's experts on flies, they would have a hard time telling me that this is a fly. You cannot calculate a fly out of this. And what I'm going to try to convince you of today is that the reason you cannot do that is because this description is missing information about space, about time, about concentrations. So in my lab, we're trying to endow each one of these arrows with molecular and quantitative information that allows us to reach a predictive understanding of developmental decision making. Basically, could you predict the fruit fly patterns from knowledge of this network topology? To put this in other words, what we want to do is to know the output pattern of gene expression from knowledge of the distribution, spatial distribution of input activators and repressors, and the arrangement of those activators and repressors on the DNA. Basically, we want to be able to predict input-output functions. Just like when I draw an electronic circuit diagram, I can go to the Horowitz and Hill book and precisely predict output current as a function of input voltage without doing an experiment. I want to be able to look at DNA sequence and predict these input-output functions. And I want to do it so well that I can predict what will change as I tune the number, the placement, and affinity of binding sites for the different activators and repressors on the DNA. Okay? All right. So hopefully, I give you a little bit of a sense of the types of questions that we're trying to answer. And now what I'm going to do is talk to you about what I call the standard model of cellular decision making, which is rooted in equilibrium statistical mechanics. Okay, and this is going to be the main topic throughout the talk. And the work that I'm going to show you today was spearheaded by Liz and Jonathan in the lab. I think Jonathan is here. I don't know if Liz is here. And it was done also in collaboration with my friend Shelby, who's on the faculty at Northwestern. And I keep talking about statistical mechanics. And, of course, we know that cells are, if they were in equilibrium, they would be dead. But there are some reasons to believe that maybe you can use statistical mechanics. One of the reasons is that, as you will see, the molecular processes that we're trying to describe have energy scales that are comparable to the thermal background. We're going to talk about DNA opening and closing, being wrapped and unwrapped. That's worth 20 kTs. Binding and unbinding of activators and polymerases is 10 kTs. Interaction energies are 3 kTs. Ion channels that drive our brains, opening and closing is 1 kT. Okay, so the energy scales are right. Maybe if there's the right separation of time scales, we can still invoke equilibrium. Okay, and Eugene Wigner wrote about the unreasonable effectiveness of mathematical models in the natural sciences, and these statistical mechanical models have been unreasonably effective, at least in the context of how bacteria make decisions. Okay, so let me illustrate that with an example from my PhD, where we looked at how bacteria decide to make a gene based on a repressor binding to the DNA and not letting the Xerox machine bind to the DNA. We used that mech to predict the level of gene expression, how much of the gene is made, as a function of the number of repressors, where each curve here corresponds to a different node, like the binding energy of repressor to the DNA, for example, or the number of genes. Like it's not the same to have 100 genes and 1 repressor than to have 1 gene and 100 repressors. The point is that all these curves existed before we took the data. There are no free parameters here, and then we went and took the data. And what you see is that to a large extent, we could predict from DNA sequence the input-output function. Not only that, all these curves look very different, but that's because we didn't plot the right x-axis. You can do some math to show that there is a quantity that you can define as the effective repressor number, in which case all the data collapses onto the same master plot, which shows you that we found the natural variable of the system. So the point I want to make is that at least when we try StatMech in one example, it worked beautifully. And the question that drives my lab in big part is, can we pull this off in the context of an animal? Okay, so what are we going to do? We're going to say that the more likely it is to find the polymerase Xerox machine bound to the DNA, the more often it's going to start making RNA, the more protein I'm going to get, such that the rate of RNA production is proportional to the probability of finding RNA polymerase bound to the DNA. And to calculate this p-bound, we're going to use StatMech. We're going to, and as you know, if once you know that you're in StatMech, there's a very clear protocol. I need to first identify the relevant states or configurations that my system can be. For example, the promoter landing site can be either empty or occupied by the polymerase Xerox machine. Then I need to figure out what the energy are of these states. I need to figure out the multiplicities, and that allows me to compute the Boltzmann weights. And then I can calculate the probability of the state I care about by dividing the weight of that state by the sum of all possible states. Okay. So let's see if I can use this very simple protocol, not in the context of predicting the whole fruit fly, but what if we look at an individual error? What if we look, for example, how this activator bicoid activates this gene called hunchback? And the names don't matter too much. You might have to remember bicoid, but I'll keep reminding you of it. Bicoid is an activator that is distributed along the anterior posterior axis of the embryo in an exponential grid. And there's a beautiful story here of reaction diffusion that we can talk about later. Bicoid binds to 6 sites on the DNA and in the vicinity of hunchback, and that leads to the production of hunchback in a step-like fashion. Okay, so this is the system I want to model with StatMech. And what I'm going to do is start by enumerating the states that I care about. Imagine there's only one binding site for bicoid, so I can have the DNA empty, occupied by the polymerase Xerox machine, occupied by bicoid, or bicoid and activator and polymerase can be bound at the same time, and then I would have an interaction. Okay, so far so good. But one detail I need to point out is that DNA in the cell does not exist in this nice linear fashion. Why? Because remember, you got like 2 meters worth of DNA, and these 2 meters need to be packed in a nucleus that is on the order of 5 micrometers. So how does a cell fit all this DNA in there? Well, it packs the DNA into increasingly compact structures. And one such structure is called the nucleosome, where you can see that the DNA is wrapped around these little circles. Okay, and this wouldn't be relevant at all for this talk if it wasn't for the fact that we know that if DNA is wrapped around a nucleosome, activator proteins cannot bind. If DNA is unwrapped, then the activator proteins can bind. So this is an extra complication, but it can be fit just fine in the context of our thermodynamic framework, because all we're going to say is that you pay some energy to open the DNA, and when the DNA is open, now the activator can bind. Okay, which means that I can add just one state to my system where the DNA is wrapped, and then the other states corresponding to the unwrapped DNA. Okay, I need to tell you one more thing, and that one more thing is that the fly can regulate the accessibility of the DNA, can regulate the equilibrium between unwrapped and wrapped. And it does so because there's another protein called Zelda that can bind to the DNA. The cartoon model is that Zelda doesn't interact with anybody but the DNA, but it's really good at binding DNA, such that when the DNA is bound, Zelda binds there and locks the DNA in the open position so that it's easier for the activator to bind to the DNA now. Okay, so Zelda and bicoid are not talking to each other directly, but they're interacting through the allosteric change in the conformation of the DNA. Once again, I can add more states to my model. We're done now. We got bicoid, we got Zelda, we got interaction energies. And what I can do is go through the StatMech protocol and come up with the different statistical weights that correspond to each one of these states. Okay. And I don't want to go through the math here, but let me just say that these weights allow you to identify the knobs that you could think of tuning. You could think of tuning the DNA unwrapping energy, the number of activator molecules, the number of Zelda molecules, binding energies, and such. But the point is that once you choose a setting for each one of these knobs, once you choose a set of parameters, you're done. Because you can calculate now the input-output function, the output rate of polymerase loading as a function of the input concentration of Zelda and of bicoid. Okay. Basically, what I'm telling you is that once I have these parameters, I'm done. I'm ready to test the model as long as I can measure the rate of production and the concentration of the 2 input proteins. Okay. All right. Okay, so I just gave you a little sense of this standard model and how it was successful in bacteria and how we're trying to see whether it can work in animals. And this led to a prediction that we need to test experimentally. And as I'll show you in a second, it turns out nobody had the technology to test these predictions. So we had to invent it. Okay. So specifically, I just walked you through the standard thermodynamic model. There are other models. We've played with non-equilibrium models. We and others have done this. People have also thought of development in the context of dynamical systems. But what all these models have in common is that ultimately they all predict an input-output function. They predict the output rate of RNA production as a function of the concentration of the input activator in single cells. And this is all fine, but we had a lot of models and no technology to actually measure these input-output functions. So lots of models without any power to test their predictions. Okay. And let me show you just a little bit of a sense of why we couldn't do this. What was missing in the technology? Typically, for example, let's say that you want to measure the dynamics of your input activator. Typically, what you would do is grab the gene for the activator and add a sequence that encodes for green fluorescent protein. This is the protein that we use to light up the nuclei, the cells, in the first movie that I showed. Okay, this works like a charm in when you work with single cells. But the problem is that in the main organisms, animals that we use to study development in worms and fish and frogs and flies, when you make the GFP, the green fluorescent protein, this is not fluorescent yet. It needs to mature. There's an oxidation step that takes on the order of 40 minutes. And this wouldn't be a big deal if it wasn't for the fact that the proteins that we want to look at live for only 10 minutes. What I'm telling you is that by the time the GFP becomes fluorescent, the action is over. Okay? So because of this technical limitation, people were constrained to studying dead embryos, meaning they would collect embryos from mothers that laid their eggs at different time points in development, and they would kill them and stain them with antibodies. Okay, you can see these. This is a pattern that was stained at different time points. And then they would use smart arguments to make a movie where you morph through images of dead embryos. Okay, so this is amazing. But if you want to measure something as simple as a rate of production in a single cell, you can't do this because you killed your embryo in your first time. Okay, so over the last few years, we have systematically developed technology to be able to measure both inputs and outputs. And I'm not going to walk you through all of this right now, but let me just give you a sense of how we, for example, measure the output rate of mRNA production in single cells. Okay, so here's what we do. We add a little sequence to the gene. Okay, then when the RNA polymerase makes a copy of it in the form of RNA, instead of being linear RNA, now it has a little loop. This loop is recognized by another protein that we add in the flag. And this protein is fused to GFP. Okay, note that we're not making new GFP, the GFP is already existing. So the result is that in every cell, in every nucleus in your embryo, you're going to have a little spot of fluorescence. And the fluorescence of that spot is going to be proportional to how many polymerase Xerox machines are making a copy of the gene. This is what it looks like when you put it all together. In green, you see the RNA signal, and in red, you see the DNA. Okay, you see we're at a step of expression. And just to give you a sense, this is the first time anybody can measure the process of transcription, making a copy of the RNA or the DNA in real time in single cells of a multicellular organism. And we can go beyond these pretty movies and count the number of polymerases making a copy of the DNA as a function of time. And this data reveals, for example, how this process of transcription is regulated in these bursts corresponding to the division cycles, and also in space, because you see blue is to the left of the boundary where I have a lot of green, and red is to the right of the boundary where I don't have that much green. Okay. So we have developed this and many other technologies, and we've used them to make measurements, make precision measurements that nobody could do before. Okay, so I told you we've developed technology to measure for the first time the fast concentration dynamics of the activators and repressors in the cell. We've also discovered that these activators and repressors are not uniformly distributed throughout the cell. They're present in little clusters, and those clusters seem to play a role in driving cellular decision making. We've shown that we can detect the binding of individual activators and repressor molecules on the DNA. We can measure the loading rate of RNA polymerase Xerox machine. We showed that the RNA polymerase Xerox machine is not loaded continuously, but in bursts. And we developed technologies to infer how these bursts originate from the fact that the promoter landing site fluctuates between an off and an on state. We've been able to measure the rate with which these polymerases move down the gene, the rate with which they fall off the gene. We discovered an unexpected coupling between these two quantities. We discovered bursts of protein production that are concomitant with these bursts of transcription, and we were able to measure protein degradation in single cells. And the point I'm trying to make here is that every time we measured something that people could measure before, we get the same result. But these technologies have enabled the measurements that we couldn't do before in any way. And they have fueled my research program, as I'll show you in the next few slides. But not only that, when I moved to Princeton, I met up with Lyman Page, who was the chair of the department. And you probably know Lyman. He's a great guy, very smart guy, but he doesn't know a lot of biology. So the thing that he latched onto that I thought was a great point was, this is something everybody will be using in the next 5 years. And I really took that as a challenge. And that challenge was to make these technologies available as a means to create a community of quantitative and physically minded developmental biologists. And the cool thing is that in the last few years, these technologies have now become the standard in the quantitative study of flight development. But not only that, we, with collaborators and others, have implemented them in all sorts of different organisms, from plants, fish, worms, to embryonic stem cells. And many of these have now led to papers. Okay, so once again, these technologies are really enabling quantitative biology writ large. All right, so I should show you how we invented technology to test the predictions from our standard model. So let's see what happens when we compare theory and experiment. Okay, specifically, remember, I showed you how we measure the rate of RNA production. Now I need to compare that. I need to measure the input concentration of zelda and the bicoid activator. Okay, and just like I showed you that I can look at the fluorescence of these nuclei, I can look at the fluorescence of the bicoid activator and zelda, and this fluorescence reports on the concentration as a function of space and time of these two proteins. Okay, I can plug in this information into my model, and I can choose a set of parameters, and that predicts the number of RNA polymerase Xerox machines making a copy of the gene as a function of time. Okay, and this predicted shape of the data looks, at least qualitatively, like the data that we get. Okay, so but in order to quantitatively compare theory and experiment, we're going to extract two different quantities from our data. First, we're going to measure the rate with which RNA polymerase Xerox machine is loaded onto the gene. That is given by this initial slope of the data. And then we're also going to measure the onset time, the transcriptional onset time, which is when did you start loading polymerase Xerox machines onto the gene. That is given by the EX-intercept of the time. Okay, so what I'm going to do is move along the embryo, and at each position ask, what's the rate of RNA polymerase Xerox machine loading, and what's the onset time? And this is what we did. Okay, so on the top, you see the rate of polymerase loading as a function of position along the embryo. And just like we would expect for a step, you see that the rate of polymerase loading is modulated in a step-like fashion. Okay, the onset time is relatively uniform throughout the embryo. All cells start making RNA more or less at the same time, around 4 minutes into the development. Okay, we tried to fit our thermodynamic model, and it worked great. So you could have said, all right, we're happy, we can pack our bags and go home. But then we started worrying, there's a lot of parameters here. Could we be right for the wrong reasons? Can we think of further ways to challenge our model? And that's what led us to think about a knob that we could tune, in this case, a switch. It turns out that everything I've shown you so far has both bicoid and zelda in the embryos. But you can actually make embryos that don't have zelda. You can turn off zelda, okay, and see what happens to the dynamics. And in principle, it's a very small change to my model. I just get rid of one parameter. Okay, so if it worked for so many parameters, maybe it would work for fewer parameters. Okay, so let me show you what the data looks like. On the left, I'm showing you the so-called wild type embryo that has both bicoid and zelda. On the right, I'm showing you a mutant embryo that has only bicoid but no zelda. I'm not going to show you the little dots. I'm going to color the nuclei when I see the dots, so you can get a better sense of what's happening. So check it out. On the left, you saw how every cell turns on, and it does so at the same time, more or less. On the right, you saw how not all cells turn on, and they don't do so at the same time. Okay, let me play it again. Everybody turns on the left, some of them turn off, and with a delay on the right. Okay, so once again, we move around along the embryo in an embryo that doesn't have zelda, and we plot the results. First, we see that not every nucleus, not every cell turns on. Only cells in this range of the embryo turn on. The cells that turn on in this region load RNA polymerase Xerox machines more or less at the same rate than the ones from the wild type embryo that have zelda. So you couldn't tell them apart from this measurement. The interesting thing that we discovered is that there was a change in the timing of when these cells turn on. And this is a very small change, right? It's just a few minutes. There's no way you would see this by killing embryos and fixing them, okay? But our live imaging can reveal those. I told you before that the standard thermodynamic model worked great when I had Zelda, but we tried to systematically scan parameter space and thermodynamic model space, and we couldn't find a single set of parameters that could account for the mutant data. We couldn't account for the fact that there's a delay in the on time as a function of position along the end. Okay? And why is that? Let me give you a little bit of an intuition for this. Okay? So let's just take a step back and think of when are we allowed to use equilibrium? What I'm showing you here is a kinetic version of our model where you see that the DNA can turn on, open and close. When it's open, you can have an activator bind, RNA polymerase Xerox machine bind, and then you can make RNA. In order to be able to forget about the transition rates and invoke equilibrium, I need, one of the key assumptions that I need is that the dynamics of the input activator have to be much slower than the dynamics of binding and unbinding of the activator and the dynamics of opening and closing of the DNA. Also, the dynamics of making an RNA, of initiating the process of transcription, have to be much slower than the same process of binding and unbinding and opening and closing of the DNA. Okay, so I need this separation of time scales. And separation of time scales imply that there's an instantaneous input-output function, that the instantaneous concentration of the input B-code activator dictates the instantaneous loading of RNA polymerase Xerox machine. Okay? What does that mean? Well, imagine that I go to one position along the embryo, and I measure the B-code concentration as a function of time. I'm going to have dynamics that look like this. And you can see that the input-output function, if it's sharp enough, it defines a threshold. If B-code is below this threshold, if B-code concentration is below this threshold, I don't get production of RNA. If B-code concentration is above this threshold, I get production, right? And you see how the B-code concentration crosses that threshold for some time during development. What this tells us is that when B-code concentration crosses that threshold, if you're able to measure the number of RNA polymerase molecules making a copy of the gene, that's when you're going to start seeing fluorescence. So this threshold defines when do I start seeing the little bright spots. Okay, but the one way to look at our data is that our data does not respect this instantaneous input-output function. Okay, and let me show you an example. Here, once again, I'm showing you B-code concentration as a function of time for a given position along the embryo. There's a threshold here that defines when I start seeing fluorescence, as shown in the output plot for the number of RNA polymerase machines making a copy of the gene as a function of time. Okay, I'm gonna go now to a different position along the embryo. B-code, remember, it decays exponentially as you move along the embryo. So now you got dynamics that are whose amplitude is lower. But there's a point where B-code will cross that threshold. So I would predict that I should start seeing fluorescence at this time point in development. But what we see is that the process of transcription, of making a copy of the RNA, begins much later than predicted by the threshold. And this is true along the whole embryo. There is no unique threshold along the embryo, okay, which tells us that this fundamental assumption of separation of timescales is not working here. All right. Okay, so I showed you that we took this cartoon model of how decisions are being made seriously. We turn it into a mathematical statement. We challenge the model by tuning one of these parameters, and the precision measurements enabled by our technologies reveal that there's no way the standard thermodynamic model can recapitulate the data that we observe. This tells us then that, you know, we did a full round of the theory experiment dialogue, and what we need to do now is go back to our theoretical model and try to revisit the assumptions. Okay. In particular, what we're going to do, I'm going to show you how we sacrifice the assumption of detailed balance and equilibrium. In one step of our model, we can recapitulate all the data. Okay. And specifically, just let me remind you that what I'm after is a model that can explain why, when I have both B-code and Zelda, every cell turns on at the same time. But when I don't have Zelda, cells turn on with a delay, depending on where they are along the embryo. Okay. One simple mathematical model that could explain this is if you sacrifice the reversibility of opening and closing the DNA. Let's say that you had an irreversible transition from closed to open, from inaccessible to accessible, and that the rate of that transition depended on the position along the embryo. Okay? If this is true, then I could calculate the rate of change in the probability of being in the open state with this simple rate ODE that I'm showing you here. We're going to then posit then that once the DNA is open, now it's locked in the open configuration, and now activators and repressors can bind to the DNA in a thermodynamic fashion. Okay, so now I'm still going to describe the probability of finding RNA polymerase bound to the promoter landing site using Boltzmann. And in this case, the total rate of production of the RNA is given by this formula here, where you see that there's a position-dependent delay in the transcriptional onset time. So basically what I'm advocating for is like trying to find a way to revise our model and use this as a microscope to figure out what is the mechanism, what is the type of mechanism that could explain my data. Okay. All right. So I need to figure out, who could be driving this position-dependent delay. Okay. And once again, I got a delay as a function of position. And just let me remind you that Bitcoin, the one molecular player, the activator that is feeding into this system, varies with position. So what if it was Bitcoin that not only activates recruits RNA polymerase Xerox machine to the gene, but also catalyzes the opening of the DNA so that it becomes accessible? What I'm advocating for is a different model from the equilibrium model of DNA accessibility. In the equilibrium model of DNA accessibility, remember, you got the DNA opening and closing due to thermal fluctuations, and Bitcoin is just a passive bystander in this DNA accessibility game. Only when it's open, it can bind and do its thing. It turns out that there's biochemical evidence that suggests that Bitcoin activator and other activators could be using energy to irreversibly catalyze the opening of the DNA. Okay, so and they do so by, for example, consuming ATP, which is the energy currency of the cell. So the idea then is that Bitcoin catalyzes the opening of the DNA. It's now irreversibly open, and now you can play the equilibrium game again. Now Bitcoin can bind and unbind, and you can describe its binding through equilibrium. Okay, so that's the model that we're positing. And note that we just sacrifice beta balance in like one little part of our model. And this model works pretty well. Okay, so here I'm showing you the rate of polymerase loading as a function of position along the embryo. Okay, and what you see is that I have the wild type and the Zelda mutant, and you see how it does pretty well. Okay, so just like the thermodynamic model. The cool thing is that this model does pretty well at capturing the position-dependent delay in the absence of cell. Sure, it predicts that you should see cells that make RNA a little bit further into the embryo. So there are some things that we need to think hard about. Okay, but I would say this is a good amount of progress. All right. Okay. So we're very excited about this model. And I'm going to tell you a little bit more about it in just a minute, but let me just take a step back. Let me remind everybody that what we were after here is being able to understand and predict cellular decision making so well that I can predict input-output functions from knowledge of the arrangement of binding sites for activators and repressors on the DNA. Okay, and I showed you that to do this, we had to account not only for the binding and unbinding, but also for the opening and closing of the DNA. We started with a cartoon model, the type of model that you would find when you just get started and look at the phenomenology. We turned that into a precise mathematical statement by positing that this process can be described by equilibrium statistical mechanics. This made predictions about the rate of RNA polymerase loading as a function of the input concentration of Zelda and Bitcoin. To test the predictions of these models, we had to invent new technology to measure both inputs and output. These technologies didn't exist, and we had to invent them. Okay, and these enabled the precision measurements necessary to test them. This allowed for a dialogue between theory and experiment that we challenged by turning a knob, that is that of getting rid of Zelda. We had two input players, we got rid of one. It should have made life easier, it made life harder, because we discovered that we couldn't explain the dynamics resulting from the regulation from our model using equilibrium. And this led to a discovery that if you invoke non-equilibrium, a non-equilibrium or an irreversible transition that is catalyzed by your activator, you can going from inaccessible to accessible of the DNA. You can pretty much recapitulate all of the data in a quantitative fashion. And of course, now this is just the beginning of a new cycle of theory and experiment. How do you probe this model, this and other alternative non-equilibrium models? Okay, and I started this talk by saying I know that life is not in equilibrium, and everybody knows that. Yet, equilibrium models are amazingly good at describing nature. But we and others have been trying to figure out when does the equilibrium break, and they're having many challenges in figuring this out. One of the challenges that we really don't know where this energy is being dissipated. Like it's been challenging to figure out what steps of, let's say, the simple model, and what steps of the simple model are you consuming energy. Not only that, we don't know if that energy is being used for something useful. Okay, for example, one signature that you could see is that when you use energy, instead of having a relatively shallow input-output function, you get a much sharper input-output function. Okay, so we and others have been on the lookout for signatures of non-equilibrium processes in cellular decision making, and maybe we found one here that was only revealed by looking at the dynamics of when the process of transcription turns on. So how do we go about answering these questions and probing our models? Well, one of the challenges we're facing is that reality is too complex. What do I mean by this? The cartoon models I showed you before today, had like only one B-code activator binding site that interacts with the RNA polymerase Xerox machine. But reality is a lot worse because you have multiple binding sites for B-code activator, and you just gotta there's an explosion of parameters because you have on the order of 6 binding energies, in this case at least, 15 B-code interaction energies, 6 B-code RNA polymerase Xerox machine interaction energies. This gives you a model with at least 27 free parameters. And, I don't know about you, but I'm not in the business of trying to fit a 27 parameter model to the simple input-output functions that I showed you before. So what are we trying to do? We're very inspired. Oops. There you go. We're very inspired by, for example, the work from our colleague Mike Cromie, that in what I would call synthetic quantum mechanics. I mean, you might know this work already, but he did this cool thing of creating basically almost like a particle in a box. Okay, and this is not a hydrogen atom, but it's something much simpler that allows us to test our very fundamental understanding. Okay, similarly, we want to use synthetic biology to make nature simpler, to bend nature to understand. What do I mean by this? Instead of looking at a 6 binding site DNA sequence, I'm going to create a sequence that has only 1 binding site. And I know that this sequence does not exist in nature. But I don't care, because if I don't understand how this guy works, I have very little hope of understanding how 6 binding sites work together. So the idea is to be able to predict how 1 binding site works, how the input-output functions coming from 1 binding site, how these input-output functions change as they change the binding site position and affinity. And once I understand that, I can start tuning the knob of binding site number. I can start increasing complexity in order to, at some point, get to the description of the sequences that I see in nature. And we've been making great progress along this lines. I'm not going to walk you through this, but what I'm showing you here are the results of probing the 2 aspects of our model, the irreversible transition from closed to open, and the thermodynamic binding and unbinding of activators and repressors. You can see data here that has to do with testing of each one of these models in the context of synthetic DNA sequences, where different curves correspond to tuning the number, the placement, and the affinity of repressor and activator binding sites. And we're very excited to keep going down this path. All right, I'm almost done here. Let me just remind you that I told you we need to endow these networks with quantitative information, with predictive information, and there were challenges along the way. We needed technology to measure input-output functions. You needed to know something about the cartoon model so you can turn it into a mathematical model, and you needed to figure out how to put this all together to derive and test theoretical models of cellular decision making. And this represents the three main threads in the lab that I walk you through today. We developed technology to turn the fruit fly into substrate for precision measurements. We use physics as a microscope to rewrite the cartoon models of the molecular mechanism underlying cellular decision making. And we use all of this to put together what I call physical biology of living embryos. And even though I showed you one example of this dialogue between theory and experiment, there's a wide repertoire of other cellular decisions that we have dissected, and I invite you to ask in the questions, or we can talk later about this. Okay. All right. So I told you that this work was spearheaded by Liz and Jonathan in the lab with the help of Shelby and also Melissa, a collaborator at Wisconsin. But this is not just the result of a few grad students. It's just amazing the stuff that we've been able to get done, just because we've been surrounded by this amazing set of students and postdocs that are willing to take risks and do crazy stuff. And they are really the ones that should be acknowledged. So, I'm just so grateful to everybody in my lab over the years. It's just great. And that's one of the fantastic things about being at Berkeley, how easy it is to surround yourself with people that are way smarter than you. But also, putting these pictures led me to the realization of how much of a living organism a lab is. And there's no one picture that features everybody. So this is, if we were all in person, I would ask them to stand up and you guys could applaud them, but maybe you can do the Zoom applause for them, because these are the people that have really been driving the research that I've shown you before, that I've shown you today. I also need to acknowledge the great biophysics group within physics. It's a great, it's been such a wonderful set of people, and official and unofficial mentors. Also, some of my buddies in all the parts of the physics department. When I joined here, I set myself as a homework to take everybody out to lunch, and the department is so big that I'm not done, but I look forward to taking people out to lunch once we're back to some sense of reality. I also need to acknowledge my co-authors from the book Physical Biology of the Cell. I've learned so much from them, and words can't describe how supportive they've been. Let me also acknowledge our funding sources that give us the freedom to ask these crazy questions and do what we're most excited about. And finally, and I think this probably will hit home for anybody having kids at home during these 2 years of pandemic, let me acknowledge my family and my wife that has been literally juggling, as you can see here, the 2 kids and me being stressed out. And, it's been so supportive, and I wouldn't be anywhere close to where I am now if it wasn't for them. So with that, thank you very much for your time and your attention, and I'll be very happy to take any questions you have.
Thank you very much, Hernan, for this wonderful talk. Table is open for questions. I see a couple of people already raised their hands. Let me start with Aditya. Please unmute.
Oh yeah, sorry. I was clapping, but I'm very happy to ask a question. I had a question really about I guess Hernan, you spoke about the non-equilibrium inaccessible state. So is the claim then that once this speakoid binds, the DNA can't be wrapped back around?
At least, yeah, unless somebody, like in the model, at least somebody else interferes now to block the DNA in the closed configuration, and we know of precursors that do that.
Okay, that's interesting. I guess the other thing was
not on the scales of this experiment, of this particular experiment.
Can you also? I was a little bit confused by why equilibrium requires a separation of timescales. Could you comment on that a little?
Sure. Let me find the slide. Well, I mean, basically, if I want to be able to, invoke Boltzmann weights, I need enough time for the activators and RNA polymerase and the opening and closing to explore the different states, right? So, if for a quasi-equilibrium to be able to be invoked, I need the binding and a binding to occur a bunch of times, such that it's averaged by Boltzmann, and that can only be done if all the things in the system change on a timescale that is much slower. So maybe basically when you go from here to here, the system has bound and unbound a bunch of times so that you don't need to worry about the transitions, but you just need to worry about the relative energy levels. Does that make sense? Like basically I'm trying to, you need to invoke quasi equilibrium. And, clearly nothing is in equilibrium in biology. So you need to, only in the right regime of separation of timescales, you can use that.
Thanks.
Sure.
Okay, good. Yeah, Eli Jablonovic has also a question.
Yes, very fascinating topic. I've been interested in it for a long time. Now, as I see it, there is a string of digital data that's in the DNA. And from that linear string of digital data, I'm supposed to get an animal with shapes and all sorts of wonderful things.
Yes.
Now, so that's the challenge, is how to go from one to the other. At the very least, one can visualize maybe some nonlinear mechanism or some little machine that reads out the digital data. And so is this what you're looking for, is the details of that machine?
Well, I just want enough details that allow me to make predictions. I don't, there are some details that I don't care about. Like, we're just after the right degree of coarse graining. Okay. So, at this point, if I can figure out how, imagine that you could define a basis set for development. I just want to understand how you make gradients, steps, and strengths. And maybe after that, maybe I can figure out how they all get put together to make an embryo. And maybe after that, I can understand how the physiology and morphology is encoded. So yeah, I care about molecular mechanism insofar it allows me to write quantitative predictions about what these arrows are. Does that answer your question?
Yes. So where are you in this progression? You started at the top right, you ended up at the bottom left. How far did you get?
Well, I would say that I got to the point where we have now a relatively good understanding of how you make a step. Okay, but I would say that even there, I still can't predict what will happen if I change the number of binding sites in the vicinity of the gene that encodes for that step. And that's why we're now engaged in this effort in synthetic developmental biology that I was introducing you to. I think, we revealed that the standard model doesn't work, but now to really probe the model, the predictive power of the model, and to refine it, I think we need to go to the synthetic platforms. And we're starting to make progress on that. That's probably one of the things I'm most excited about doing next.
Yeah.
In this.
So that would be a real prediction. You synthesize a digital sequence and you get something different.
Absolutely. Absolutely. And that's exactly what we've done here, which, I didn't walk in detail, but, this, for example, corresponds to having one precursor binding site and putting it in different positions. And most of these curves still have a few free parameters. So they're not parameter free like the ones I showed you for bacteria, but we're almost there. We have some experiments now that if we understand this, I think we should be able to make parameter free predictions in the next round of experimentation.
Okay, thank you.
Sure.
Great. Table is open for questions. I'm sure that there are plenty of questions. At least there are plenty of open research questions. David.
Hi, thanks, Hernan. It was a beautiful talk. It's really impressive to see the ability to spatially and temporally resolve all of these measurements. So I had maybe two related questions concerning rolling out kind of equilibrium models. The first is the role of noise. So, having simple reaction diffusion equations also implies some sort of mean behavior. There are fluctuations that can cause deviations from kind of the simple first order generalization kinds of equations. So I'm wondering how much that would play a role, if you can really ignore that. And the related question is, so I guess your discussion of equilibrium as being a timescale separation, I think is certainly true, but another aspect of actually evolving to a Boltzmann distribution is the non-existence of cycles. So if you have cycles or persistence currents that goes through the states, then you could evolve to a steady state, the one that doesn't change in time, but still not be a Boltzmann state. So I wonder how much that sort of physics plays a role in deviating from the Boltzmann statistics you find.
Yeah. So, okay, let me answer the first question, which is that we are trying to go beyond the averages and look at the distributions. Okay, so, everything I showed you was exactly measuring the mean onset time but we're trying also to measure the distribution of onset times. And Jonathan, who's here on the call, has done some great work trying to understand how much could you use this distribution to figure out what's going on under the hood. Like, we have reason to suspect that maybe there isn't just one off state, that you need to go through multiple off states. Like nucleosomes, for example, now we're getting technical, but have like 11 discrete contacts between the DNA and the histone protein. So you could imagine that each one of them corresponds to a little bit of a step around robin. And it gets tricky, but we're making good progress on that front. Okay. So the other point that you made is absolutely let me see, I have a nice slide for this somewhere. Where is it? Yeah, you said, hey, you might have to, it's not just about separation of timescales. You cannot sacrifice detail bounds. These cycles, the cycle in one direction should be the same as the other direction. This, linear things are a little bit trickier to put in that context. But let me say that we actually tried sacrificing detail bounds. Okay, so forget about the thing to the right. Okay, but we looked at the most at general models where you sacrifice detail balance, and even those could not explain the data. You had to invoke some catalysis of the opening step for it to take place. Okay, so we did test those models to the degree that we could. The computational exploration gets tricky, so we could only do it up to 5 binding sites, I think. But at least in our computational exploration, we could not find a model where you sacrifice detail balance that could explain the data without invoking some active role of the activators in opening the DNA. Does that answer your question?
Yeah, thank you.
Thank you.
Okay, good. There's a question in the chat. The question from Edward reads, how do synthetical models stay alive?
Well, I mean, the flies, when you make the synthetic, okay, how do the synthetic, I think what it means is that when you make a synthetic DNA sequence, how does this fly stay alive? Well, the thing is that we make these synthetic molecules and we decouple them from the rest of the network. They read out the information from the network, but their output is not being used by the fly in any way. A great challenge would be to now get rid of the actual network and hook and rewire it with synthetics. And at some point we want to do that, but we're not there yet.
Okay. Good. There are more questions. I don't see one. I actually have a question. So suppose you really develop the best technology, not the best, such a good technology that you can figure out all the mechanisms. You really have figured, okay, this is what's going on. Mm-hmm. Exactly. But in the end, you want to make a prediction. And basically, do you have a sense of how complicated of a mathematical model that would correspond to, and whether there would be a chance of actually running it through it, and how far can you get there? I mean, I guess
if you were to try to model the whole fruit fly.
Yeah, or I mean, how far? Let's say you start with the first cell. How far do you think that modeling could get you?
Well, I think it might be about coarse graining. I mean, the first thing I want to figure out is how does DNA sequence encode for these errors. But once you know that, maybe you can just forget about what's going on under the hood and plug those in as phenomenological input-output functions, and then ask how this works from the standpoint of dynamical systems. Mm-hmm. So I don't know the answer. All I know is that I like starting simple and then making it more complicated. And it would be great to understand this to the point where you can add like a wing at will, like where you can actually engineer the fly, right? There are reasons to believe that you can describe what happens here. Once you know what these input-output functions are, that you can describe this using the language of dynamical systems. And we're working on that front as well. But at this point, they're kind of separate efforts. At some point, hopefully, they will be joined. How to predict the behavior of an individual arrow and how multiple arrows come together to make an organism.
Okay, thanks. Yeah, thanks everybody.
I think I have another question.
Okay, yeah, please, Oscar.
Beautiful talk, Hernan. Regarding now the possibility of thinking about non-equilibrium models in general that seem to be realized in these input-output functions, I have the question, do you think this is kind of really significant in terms of the function that cells have to perform? For instance, Hopfield has shown that there's something like kinetic proofreading, which only works in non-equilibrium when you have non-equilibrium processes, right? So this is something that's kind of prevented when you have equilibrium constraints. So can you imagine something like that also for gene expression?
Yeah. So one that people have been focusing a lot on is whether you can get sharper input-output functions. Sorry. That with non-equilibrium than with equilibrium. But actually, Nick in the lab is thinking about, I think this is a very interesting measure, which is how quickly, like let's say that, what do neighboring cells have to do? They have to determine whether they're exposed to more or less activator, let's say, to decide whether they're going to become A or B, okay? And one of the things we've been doing is looking at the rate of information production when you're in equilibrium and out of equilibrium, and asking how quickly can cells be confident about the input concentration they're reading. And what we can see is that if you don't spend energy, you're going to take a really long time to read out the input concentration to the degree of precision that you need for cells to decide, neighboring cells to decide on different developmental phases. So yes, no, we're very much interested in that. People like Gasperg and Eric Stigia and Heinrich Berger have been thinking about these things as well. And we think we have an interesting angle on it as well that is very much inspired by kinetic proofreading by Hopfield and Nino. Absolutely. And what I think is very cool about this is that, it takes us into this realm of active, driven, living matter. So, it's just super fun.
Okay. Are there more questions? Okay, I don't see any. Last chance. Okay, then let's thank Anand for his wonderful talk. And yeah, see you all then next week.
All right. Thanks, everybody. Have a good night. Right. Guess I stopped the recording. Why? Bye, guys. Thank you.